In this module, we're going to be looking at dimensionality reduction algorithms, which in simple terms is a way of minimizing the number of variables or measurements that you have in your data in order to simplify the visualization of your samples. For this example, we're going to be using a neuroblastoma dataset consisting of 498 tumors. And the first dimensionality reduction algorithm we're going to be looking at is a relatively commonly used one known as the principal component analysis. So under the type of analysis, you can select principal component analysis and click Next. So here, we're going to leave all of the adjustable settings for now as they are and click Next one more time. And we're going to plot the PCA results. So here what you can see is that in theory, each sample has thousands of measurements per gene. And these measurements have now been minimized down to a specific number of smaller dimensions known as principal components. And here you can see we are visualizing principal component 1 compared to principal component 2 in a 2D plot. In this case, each dot on the plot represents one sample, and you can see that you can hover over the samples and again see the sample annotations that are available for that specific sample. And they are portrayed then in a 2D space separated by principal components. Now, at the moment, they're not colored based on any sort of characteristics. However, it might be useful to be able to look at specific tracks in your data. So if you scroll down to adjustable settings, now we can color by track, and in this case we're going to look at the data based on the staging system of the disease. So if we select INSS stage and click Next, we are then presented with the same plot of principal component 1 versus principal component 2, and you can see that the stage 4 tumors seem to be separated out from the other stages of the disease, which remain in the bottom left corner of the plot. Now this is a plot that has been reduced down to 2 dimensions, principal component 1 versus principal component 2. You can also look at different principal components, so you can compare component 1 versus component 3 or component 2 versus component 3, which will give you just a slightly different representation of the data. However, different principal components will explain different dimensionalities within the data and may separate samples in different ways. Another option is that you can also look at this data in the 3D space and compare principal component 1, 2, and 3 at the same time. So if we select that and click Next, we will then generate a 3D plot, and you can see here that you have then a more interactive plot on the left where you can see the separation of the samples slightly more clearly. So while here we've looked at staging and how that separates in the different groups according to principal components, it is very easy to switch between different tracks to look at perhaps other tracks of interest. So currently we're looking at stage. We can then select 𝐸N status, for example, and see what information is given to us there. And here we can see that you do have a clear separation of 𝐸N patients off to the right. And in the 3D space, you can indeed see that they are quite separated from the other patients. So here we've looked at viewing tracks on these 2D plots. However, it is also possible to look at gene expression and how certain genes are expressed in these subgroups of patients. So in order to do that, we're just going to switch back to looking at only principal component 1 and 2. And instead of coloring by track, we're now going to color by gene. And we're going to look at 𝐸N as our example here. So notice here that the 𝐸N amplified tumors were a subgroup off here to the right, which can be interesting to remember for the next plot. And then we are going to change just the color scheme for ease of viewing from a blue to red scale, and click Next. And here you can see that the higher expression is colored in red and the lower expression is colored in blue. And you can clearly see that the tumors that were segregating off on the right, which were 𝐸N amplified, indeed also have the highest expression of 𝐸N. So while principal component is a good method of dimensionality reduction and for ease of separating samples based on many observations, there are other more modern techniques that have been used, specifically developed for visualization of multidimensional data. So we're going to look at another example now. We're going to return to the main page, and we're going to look at something called tSNE. So because tSNE has the potential of being quite time-consuming and heavy on the R2 servers, depending on the number of samples in your dataset, we have pre-performed a number of tSNE analyses on the available datasets and stored the results under tSNE maps, which can be accessed in the left panel here. So if we click on tSNE maps, you can then see that we are presented with a number of already run tSNE results on many different datasets that are available in the R2 platform. So for this example, we're going to be, again, using the tumor neuroblastoma dataset consisting of 498 tumors. And you can see that it was created towards the end of December of 2020. So we select this dataset and click Next. So as with PCA, tSNE again presents the data in a two-dimensional plot where all gene expression data has been compressed down into two dimensions, dimension 1 and dimension 2. And each dot again represents one sample, which you can hover over and see sample information. Again, to better understand how the data is represented here, we can scroll down and we can color the image by track. So now we're going to select track, and we're going to color according to the INSS stage. And click Next. And once again, we do see that the stage 4 tumors primarily cluster away from the other stages. However, here we do see that you have smaller clusters of the stage 4s, which may be different from one another. So one aspect that's interesting with tSNE is that you have a adjustable setting known as perplexity, which in simplest terms is the number of points which it tries to place within one cluster. So you can see here that the current map that we're looking at is perplexity 50. However, for different size data sets, you might want to look at different perplexity values. And one easy way of getting an idea of how the data is looking is to select all the perplexities at once. And then you can click Next. And here you can see that we get a representation of the many different perplexity values that we've looked at, ranging from perplexity 5 up to 50 in this case. And you can see that the data is spread out in different patterns depending on which perplexity is used. So for this example, we're going to select perplexity 17, as I can see here that you get an interesting clustering of the stage 4 samples, not only as one cluster amongst themselves, but separation of different stage 4s. So we can simply click on this one, and it will bring up this tSNE result for perplexity 17. So as I mentioned, it is interesting that amongst the stage 4s, you do have a separation of certain stage 4 samples, some that cluster closer to the other stages, and one group of stage 4 samples that seems to separate off entirely. So if you wanted to look into this with regards to other tracks, for instance, we could go down and again color for 𝐚𝐸 status amongst the patients and click Next. And here we can see that there is a small group that clusters off by itself that are in fact the 𝐚𝐸 amplified tumors, and this corresponds with, if I switch to the previous tab, the stage 4s that cluster off on their own, suggesting that these stage 4 tumors that cluster on their own over here are, while they are aggressive disease and stage 4, they do not contain 𝐚𝐸 amplification. Perhaps this could be something of interest and you would like to look at what the difference between these patients and these patients are. And one thing with these types of maps that we have is that you are able to select subsets of the patients from within the map. And there are different ways of doing this. So for the first example, we're going to use the Select Subset tab. So if we click on that, you then see that you get a representation of the map as we saw before. And here you are able to use the lasso function and simply click and draw around subsets of the samples, which will then automatically be selected and labeled as group 1. We can then click on the second group of patients, which we know are the 𝐚𝐸 amplified stage 4, and we can select those, which will then be labeled as group 2. For instance, we also know that there are other stage 4s here, so we could make, in theory, 3 groups of stage 4 patients, so we have now selected them as such, and we can, for instance, save this or build a track within the data and then go on to perform downstream analyses such as differential expression between the groups or many other analyses that are possible. The other option for defining groups within this map is using the DB scan select option. So you can click on that. And here we make use of an approach called the DB scan, which has a couple of different parameters which you can set. And one of those parameters is called epsilon, which in simplest terms is the radius or area that samples must be contained in to identify them as a cluster. And you can also set such information as the minimum number of points that need to be within that radius in order for it to be defined as a cluster of samples. So you can see here, simply by selecting DB scan, it has automatically selected a number of subclusters within our data. And this is using an epsilon value of 3.85 and 5. However, these parameters can be changed. For instance, we can change this epsilon to 4, and we can look at a minimum number of points of 10. And if you refresh the graph, you then see that you get a different number of clusters formed. And here we can clearly see that within the stage 4 tumors that we had identified before, you can see that you do get a cluster of those which we previously know correspond to stage 4 MYC-N amplified, as well as identifying another cluster of stage 4 that are MYC-N non-amplified. You also identify a number of other clusters that may be of interest that seem to differ from the vast majority of samples in the dataset. So as I stated before, you can then save this as a track, which could then be used for downstream analyses. So why don't we go ahead and do that this time? So we'll click Save as a Track, and here you can see that each sample is then corresponding to one of the groups which you defined in this DB scan approach. So if you scroll down to the bottom, you can then define the colors that you want to associate with each of these groups, and you can save this track, which can then be used for instance, differential expression analyses, or you can use it simply for visualization of the same data later on the tSNE map. You can also choose to save this as a temporary track where it will only be saved for 24 hours, or you can store it as one of your personal tracks so that you can use it at later time points. And as we showed previously with the principal component analysis, you are also able to color this plot by gene expression, and then you can look for specific genes of interest. Again, we'll look at MYC-N in this case and select the MYC-N reporter and looking at it from a blue to red color spectrum and click Next. And here again, you see that you do get separation of the high MYC-N expressing samples that correlate with the MYC-N amplified tumors. So we've now looked at examples of both the principal component analysis and the tSNE maps for dimensionality reduction algorithms. And there is one more example that we'd like to show you, and that is UMAP, otherwise known as Uniform Manifold Approximation and Projection algorithm. So to access that, we're going to go to the tSNE maps in the left column. For the time being, the UMAPs are accessed under the tSNE map drop-down box. However, we are working on making the UMAP a separate analysis altogether. So in order to access this one example that we're going to use is from the cell line, the CCLE, the Cancer Cell Line Encyclopedia, consisting of 917 different cancer cell lines. So if we select this one, you'll notice here that the transformation is listed as UMAP. So we're going to click Next. So as with tSNE, we're presented with a map of two different dimensions, dimension 1 and dimension 2, and each dot represents one sample. You can see here that the samples are slightly more segregated than in the previous maps that we've looked at. This is partially due to the algorithm, but also due to the fact that we're now looking at different cell lines that differ from each other very much. If we scroll down to the adjustable settings, you can see here that under perplexity, you have the different UMAP settings. And here we set both the nearest neighbor number, so the number of samples that should be within a cluster, and the minimum distance, so how close together the samples must be within a cluster. So these settings you can then select to define how concentrated or spread out the clusters are, and you can play around with these to try to find your ultimate combination. So for this example, we're going to leave it as having the nearest neighbors of 30 and the minimum distance of 0.99. Just to give us a little bit more information about the plot, we're then going to color it by track, and we're going to look at the histology of the samples. And click Next. So what you can see here is, just as in tSNE, you do get multiple clusters of different samples. And here what we can see is that you seem to have a large separation of the samples on the right side compared to the left side. And if you hover over these, you can see that the ones on the right include hematopoietic neoplasms, lymphoid neoplasms. So we're separating the blood samples on the right from other types of malignancies on the left. For instance, the carcinomas, as well as you have groupings of neuroblastomas with Ewing sarcoma, as well as rhabdomyosarcomas. This does show that the samples do seem to cluster based on phenotype of the type of cancer that they are derived from. To further investigate, for instance, within the carcinomas, which seems to be quite a large group, we can, of course, select then a subtype of histology to get a little bit more definition on the samples. And again, here you can see that according to subtype, they seem to cluster more together within their individual subtypes, just to give a little bit more information about the samples in this analysis. So one important difference to mention here between UMAP and tSNE is that on tSNE plots, while the information of how close the samples are within a cluster does provide you with information of how similar the samples are to each other, the distance between clusters in a tSNE map does not tell you how different the clusters are from one another. However, in UMAP analyses, the location within the plot of the clusters is important. So if the clusters are further away from each other, that does imply that they differ from each other more greatly than the information you can extract from a tSNE plot.